My name is Olivia Piacentini. It is June 27th, 2024. Pause to read case 3. Pause to read the patient blood sample values and urinalysis. Gout is an inflammatory condition that occurs causing pain and swelling in the joints. Flares begin in the lower extremities. Gout is caused by hyperuricemia or increased levels of uric acid. Increased uric acid is released as an end product in purine degradation. There are many mechanisms through purine degradation that can cause gout, such as decreased salvage of purine, decreased urinary excretion of uric acid, and overproduction of purines. Purine degradation allows for nitrogen contained in nucleotides to be degraded in a manner allowing for proper disposal. This process occurs by dismantling the nucleotide structures, sugar, phosphate, and base. The base is excreted as uric acid, which is the end product in purine degradation. For example, adenosine can be deaminated into inosine and then converted into respective base by nucleotide phosphorylase. Inosine and guanine can be further oxidized into hypoxanthine and xanthine. Particularly, xanthine is further oxidized to produce uric acid. Uric acid can be excreted in the urine, although uric acid is not highly soluble and can precipitate in high levels, causing gout. A method of gout caused by overproduction of purines is through increased production of PRPP synthetase. PRPP is a key component in purines and is created from ribose 5-phosphate and ATP. The formation of PRPP is catalyzed by the enzyme PRPP synthetase. Overproduction of purines through increased activity of PRPP synthetase due to a genetic mutation is a possible cause of gout. An increased transcription of PRPP synthetase can lead to the overproduction of the enzyme protein. Through the overproduction of purines, this leads to an increased end product in the degradation process. Through degrading more purines, more uric acid is produced. This dysfunction leads to high levels of uric acid and can lead to gout. Although increased plasma urate is expected through this process, it is unlikely to be the mechanism affecting the patient, as our patient does not exhibit elevated urinary uric acid. The next mechanism to consider causing the patient's gout is decreased salvage of purines. Purine salvage is the process in which nucleotides are recycled into purines. Enzymes that are involved in this process are adenine phosphoribose transferase and hypoxanthine guanine phosphoribose transferase, HPRT. These enzymes will combine a free base with a PRPP. A deficiency of HPRT can cause disorder in purine metabolism and the overproduction of uric acid. HPRT synthesizes IMP and GMP from purine bases, hypoxanthine, and guanine. The inability to convert these bases into monophosphates sends these bases to be converted into uric acid through xanthine oxidase. This will cause an individual to have increased plasma urate as well as increased urinary uric acid. Therefore, this cannot be the mechanism affecting our patient. Finally, this leads us to decreased urinary excretion of uric acid. As the patient revealed, he has recently increased his intake in soda and therefore fructose. Fructose is the only sugar that can raise the levels of uric acid, causing gout. Through the fructose metabolism, fructose is metabolized to fructose 1-phosphate by the enzyme fructokinase. ATP and phosphate are consumed during this process, which leads to the activation of AMP. This causes degradation into IMP, then hypoxanthine and xanthine. Finally, the degradation leads to the product uric acid. As the demand for fructose metabolism is increased, with increased fructose intake, ATP is depleted, causing a cascade of effects to produce uric acid. Fructose-induced hyperuricemia is connected to decreased urinary uric acid excretion. Reflecting on the patient's lab, this confirms the suspicion as he presented with high plasma urate and decreased urinary uric acid. Pause to read case 4. Iron deficiency anemia is one of the most common types of anemia. It occurs when there are deficient iron levels in the body. Iron is an essential aspect of hemoglobin as it is part of the heme structure that facilitates oxygen binding. However, The effect on the hematocrit is not always consistent with iron deficiency, as it does not always impair the amount of red blood cells. B6 is a cofactor for many enzymatic functions. It is essential for the transamination and decarboxylation of porphyrin synthesis. This can cause microcytic anemia, the production of smaller than average red blood cells. Therefore, this deficiency does not contribute to megaloblastic anemia. Finally, B12 deficiency is likely to be affecting the patient. Megaloblastic anemia is often caused by deficiencies in B12 and folate, two necessary cofactors for DNA synthesis. Red blood cells appear larger as there is a dysfunction in nuclear division due to impaired DNA synthesis. B12, also called cobalamin, is required for the remethylation of homocysteine to methionine and for the metabolism of methylmalonyl-CoA. Megaloblastic anemia from B12 deficiency occurs due to the decreased methionine synthesis. Purine synthesis relies on availability of methionine, as it is a source of formate. As purine synthesis is dependent on availability of amino acids that are essential, B12 deficiency will decrease purine synthesis from trapped methylfolate and inhibit methionine synthesis. Laboratory testing and results that will confirm a differential diagnosis. A CBC, also known as a complete blood count, is necessary as B12 deficiencies would reveal anemia affecting both hemoglobin and hematocrit. A mean corpuscular volume, MCV, would measure the red blood cell size to compare megaloblastic caused by B12 deficiencies. Would measure the red blood cell size to confirm megaloblastic caused by B12 deficiencies. Finally, serum B12 and folate levels will differentiate between a folic acid and a B12 deficiency.